Hello everyone. In this video, we will be discussing about the gene cloning. So, what is gene cloning and what exactly it means by gene cloning? So, we will try to answer this question by the gene cloning experiment. So, firstly, we need actually a circular DNA. So, firstly, we need a circular DNA, that is the vector, and we call it as the vector. So this is the vector, that is the circular DNA molecule. Now we also need a fragment of DNA. So this is the fragment of DNA. So here we need a fragment of the DNA. So this is the DNA fragment, and then this DNA fragment will get inserted into the vector to produce a recombinant DNA molecule. So here we get the recombinant DNA molecule. So this is the recombinant DNA molecule. Now this vector, it transports the gene. So it will transport gene into a host cell, which is usually the bacterium. So we have here the host cell, that is usually the bacterium, and sometimes other types of living cells can be used also. Now we call it as the host cell. So here we have this as the host cell. So this vector, it transports the gene into a host cell. So it will transport the gene into the host cell. So we have here this bacterium carrying the recombinant DNA molecule, and we can see here that this is actually the recombinant DNA molecule which is present in the host cell. And here the vector will try to multiply. And now we know that the multiplication of a recombinant DNA molecule will happen. So here in this step, the vector multiplies and it produces numerous identical copies. So it will produce the numerous identical copies here. So numerous identical copies will be produced here, and not only of itself but also of the gene that it carries. And then in the fourth step, the division of host cell will happen. So when the host cell divides, copies of the recombinant DNA molecule, they are passed to the progeny, and further vector replication takes place. And then after a large number of cell divisions, a colony or clone of identical host cells is produced here. So we have here the numerous cell divisions that result in a clone, and this is here the bacterial colonies which are growing on solid medium. So we say that the colony or clone of identical host cells are produced, and each cell in the clone contains one or more copies of the recombinant DNA molecule. So the gene carried by the recombinant DNA molecule is now called as the clone. So we call it as the this has been cloned. So in this way, we say that the gene has been cloned. So this is the brief introduction about the gene cloning. Hope you have enjoyed the video, and stay tuned.